Hello, my name is Seongmin and I'm from Kansas State University. Today, I'm going to talk about the role of microbial diversity in controlling greenhouse gas emissions from conserved forage. First of all, we should know about the background about my research. So we should know about what is the forage. Historically, and throughout the world, conserved forages are an integral part of raising livestock. The environment conditions for this method, the hay and silage method, is nutrient-rich ecosystems that are maintained partly or completely under anoxic conditions. Therefore, they are very good habitats for diverse microbes, which can produce and reduce the greenhouse gas. As you can see on this figure, methanogens and the denitrifiers can convert the organic matters to methane and nitrous oxide or the nitrogen. In this kind of the process. So our goal of this study is quantify the GHG emissions potentials from forage conservation and characterize the conserved forage microbiome and analyze its role in GHG emissions. For this research, we placed the 3 bays outside the Kansas beef stocker unit and monitored for GHG emissions. For the laboratory experiment, simulating silage, 648 grams of forage samples were added in 1 liter bottles to achieve the packing density of 45 pounds per cubic feet. And the moisture contents were adjusted to 70% by DI water. We taking the sample from the bay using the hay coring probe at 2 different depths, 5 to 25 centimeters and the 35 to 55 centimeters from the outside. And the DNA was extracted for the microarray analysis. And we used the GeoChip 5.0 as a functional gene microarray analysis. As you can see in my result, the methane and the nitrous oxide was produced as like the 50 ml and the 0.02 ml. And as same as like this result, we can detect some nitrous oxide from the field study of the bay. We use this kind of number and the annual production from the Kansas. We can calculate how much nitrous oxide was produced from this hay bale, and we got almost like the 300 million metric tons of the CO2 equivalent amount we can get. So we want to know what kind of things are happening in this fermentation. So we did the microanalysis. First of all, the functional genes related with the nitrogen cycle is increased in oxic and anoxic condition at first month. After 2 months of incubation, nitrogen-related genes abundances are plateaued or decreased by nutrient depletion. This kind of things are same the methane oxidation functional genes. So they increased at the first month and decreased at the second month. From this kind of result, we can get 2 conclusions. First one is the forage fermentation process produced the greenhouse gas in oxic and anoxic region, and the majority of genes involved in the transformation of nitrogenous compounds correlated positively at first month. And second, due to the depletion of the nutrient and the pH drops, methane and nitrous oxide production stops and functional gene abundance is plateaued and decreased. That's for today. Thank you so much.